Hello, my name is Jake and my partner is Ricky, and today I'll be telling you about meiosis and a little bit about mitosis. The major difference between the two is that mitosis is a form of asexual reproduction, while meiosis is a form of sexual reproduction. Meiosis and mitosis both start off the same way, with a single diploid cell. However, in meiosis, that diploid cell is divided into 4 unique haploid cells which are used in sexual reproduction. And in mitosis, that diploid cell clones itself to be an exact copy of its parent cell. This means that the person who made the picture you're looking at right now probably didn't do very well on their AP test. The cat is forming an identical copy of itself, meaning it's going through mitosis, not meiosis. However, if that cat wanted to reproduce, both cats would have to go through meiosis to produce the gametes needed to form a zygote, the first cell of a sexually reproducing organism. What we're looking at right now is a cell not currently in meiosis. The DNA is unraveled and not yet wound up into chromatids. The spindle fibers are not yet active and the nuclear membrane is still present. And this is where the magic begins. Right now, the centromeres, constituted by 2 little centrioles, migrate to opposite ends of the cell. The nuclear membrane disappears and the DNA begins to wind around itself, forming chromatids. This is when meiosis greatly differs from mitosis. The homologous pairs of chromosomes, formed by 2 sister chromatids and joined in the center by a centromere, align at the equator of the cell. They wrap around each other so tight that genetic information is swapped, and the chromatids exchange alleles, leading to genetic variability, the defining feature that sets meiosis apart from mitosis. It's this random combination of genes that makes you who you are. This happens to every set of sister chromatids, changing them into recombinant chromosomes. Now that the homologous pairs of chromosomes are aligned at the equator of the cell, the spindle fibers can attach to the kinetochores of each chromosome. They begin to pull their chromosomes to opposite sides of the cell. Cytokinesis begins, and the cleavage furrow pinches down in the center of the cell. The first stage of meiosis is complete, meiosis 1. There's now 2 daughter cells, genetically different from their parent cell. And thus begins meiosis 2. Meiosis 2 and mitosis are actually very similar because in both of the processes, 2 sister chromatids pair up, whereas in meiosis 1, homologous chromosomes pair up. At the end of meiosis 2, the cleavage furrow forms again, forming 4 unique gametes or haploid cells. In a male, meiosis forms sperm through spermagenesis, and in a female, meiosis forms eggs through oogogenesis. The nuclear membrane then reforms. Meiosis doesn't work out for every cell. Sometimes, during non-disjunction, both chromosomes of a homologous pair can migrate to the same pole, leaving one daughter cell without a chromosome. Even if meiosis 1 goes correctly, meiosis 2 can still experience non-disjunction. Both sister chromatids of a chromosome can migrate to the same side of the cell. This will lead to 2 abnormal gametes, as opposed to the 4 abnormal gametes that can happen if non-disjunction occurs in meiosis 1. Non-disjunction in meiosis 1 and meiosis 2 can result in Turner, Down, and many other genetic disorders. Humans have 23 chromosomes in their sex cells. To find out how many different combinations we can get through independent assortment, you can take 2 and times that by the 23rd power. The same goes for my example. You can take 2 and times that by the 3rd power, which is the amount of chromosomes in my example. This would give us 8 different combinations. And that concludes meiosis.